Click for IB Biology. Here we're looking at the process of morphogenesis caused by the diffusion gradients of morphogens. There's a blank PDF of this presentation down in the description, along with 10 questions, with answers of course, that you can use to check your understanding of the content. So let's frame the topic that we wish to discuss. When we look at the ball of cells from the inner cell mass, all of these cells have retained the capacity to form any part of the fetus. So that each of these cells, should we choose any of these cells, remember that each one of them contains an entire set of the human genome. So the question is, why is it that some cells become brain cells whilst other cells will become spinal cells? Others will form limbs and eyes and digestive systems. What is it that actually tells a particular cell not to express all of their genes, but just to express a select few of those genes, allowing the cell to specialize its shape and to specialize its function? The clues to this come from a process called morphogenesis, developing shape of an organism. And in fact, although this has become one of the biggest research areas in the 21st century for biology, the initial ideas for morphogenesis can be found in the writing and work of Alan Turing when he published what is called the chemical basis of morphogenesis, a mathematical paper which was published in 1952. I mention Alan Turing because, of course, he is regarded as the father of modern computers, and yet he was also speculating, hypothesizing in the area of morphogenesis and biology. So here's Turing's idea on diffusion gradients. He hypothesized that morphogens would exist. They would probably take a chemical form, and today that we know that these are what we call signal molecules, which are either proteins or lipids or indeed forms of RNA. We begin our discussion at this point here on the inner cell mass of the ball of cells, which will differentiate into the organism. And we hypothesized that one of the morphogens has been secreted in the cells here. And this means that as it diffuses outwards along this axis, we would have a high concentration at this end, and we would have a low concentration of the morphogen at this end. And so the cells from left to right in the diagram have a position on a single axis as far as the ball of cells is concerned of high to low. Now it's suggested that what's actually happening is that the signal molecules, the proteins, lipids or RNA, are adding special receptors to the cell membranes. In the second part, we have a second morphogen center down here, and there will be diffusion in this direction here. So again, we will go high to low in this direction. But the consequence of this is that every cell within the ball of cells now has a position in what we might like to think of as the XY coordinates. So in terms of this axis and this axis, each cell has a position. And finally, to establish a unique positional sense for every cell, a third diffusion takes place, beginning here in the plane and diffuses backwards in this direction. So perhaps it would help to think of this as there has been an X diffusion, a Y diffusion, and now we're creating a Z diffusion, X, Y, and Z. So 3 phases of diffusion means that within the ball of cells, the cell membranes have created a unique positional sense to each of the cells within the embryo. This means that they will uniquely respond now to the signals which will then pass through the embryo, and this will trigger their locational understanding of what to become, their positional understanding, so that where we have a brain cell, it will become it will differentiate into that particular structure. Perhaps this diagram will help to visualize what I'm saying here. So you can see that using colors, I've created unique positional senses for each of the cells in the cell mass here. And then using colors, I'm showing that cells which come from this location will then follow a pattern of development which will lead to brain tissue. The green ones are here, so they're going to lead to the differentiation of tissues around the mouth and the upper thorax, the upper tracheal areas. And then we have things like the digestive system here in orange. So those cells have that positional sense, so they know what to become. The astonishing discovery in the 21st century is that the genes which have been triggered in these different unique positions turn out to be the same set of genes. So those genes which in humans are triggering the formation of brain and spinal cord and then digestive system are the same set of genes which exist in the vast majority of organisms. And this has led to the development of a field of study called evolutionary developmental biology, which has the shortened name of Devo. And this is a very popular and massively important field in 21st century biology. And I'm just going to take a minute, which I don't normally do, to recommend reading to committed biology students, which is this book here, which is called Endless Forms Most Beautiful by Sean B. Carroll. This truly is a book for a student intending to go on and study biology or indeed medicine at a higher level. This is the definitive book which should be read to allow you to access 21st century biology. Devo, written by Sean Carroll, beautifully written, easy to read. Thoroughly recommend this book. Here is the link to the next video in the sequence, to the channel, of course, and of course, a link to the playlist.